Hi everybody, this is Mrs. Mancuso and today's lesson is about transcription. So transcription is how our cells change the message within DNA into another molecule called RNA. So I have here on the first screen, this is called the central dogma of biology. That means the central flow of information. So we start off with DNA and we know that those are large molecules that make up chromosomes and that is within the nucleus and that it doesn't leave the nucleus. But somehow our cells are able to make proteins. So what happens is specific genes are transcribed, so we're going to go through this process of transcription to produce RNA. And RNA is a much smaller sequence because it's just for one gene, whereas there could be 1,000 genes on a chromosome. So just make the RNA for a single gene, have that RNA be modified, and then leave the nucleus and go to a ribosome, and then translation, which will be our next video, will occur to produce proteins. And then proteins are what give us our traits, our metabolism, our homeostasis. So much is relying on this, so this is why we call this our central dogma. This is what everything is based on. It certainly leads into many units of biology. All right, so I wanted to show you this picture to get started. So here's the nucleus, here's a strand of DNA, here's one section of the DNA being transcribed, and that transcription produces an RNA sequence. That RNA sequence can go through modifications and become a messenger RNA transcript. That then would leave through a nuclear pore, go out into the cytoplasm, and join to a ribosome, where tRNA molecules, their anticodons, so their sequence, would match up with the sequences called codons on the messenger RNA and call for amino acids to be brought in a specific order. Amino acids, when connected, form a peptide bond, and that builds our protein. So the step in here, producing the RNA, that's transcription. That's going to be our focus today. Our next video is translation, where we talk about how these proteins are built. So let's start off with transcription. So we're talking about how RNA is produced. First of all, it happens in the nucleus. It is signaled by transcription factors in eukaryotic cells. So we've talked about signal transduction pathways and how those can turn on transcription factors that go to a specific sequence of DNA and say, hey, we're going to need this protein. There's 3 steps to transcription, and they make a lot of sense: initiation, elongation, and termination. So the first step, initiation. We need an enzyme to get this started. So the RNA polymerase is what comes in. It finds where that transcription factor is, and the region that it joins to is called the TATA box. It is a sequence TATA, and it's in the promoter region. So a promoter region is always upstream of the gene that you want to code. So right before it's saying, hey, start right here so that you can get the entire gene in. So the RNA polymerase joins to that section and then opens up the DNA, unwinds it so that it can begin reading it. All right, the next step is called elongation. So this cream-colored mass, that's my enzyme. This was my promoter sequence. There's the TATA box right there. That is where the RNA polymerase started, and now it moves in this direction. It can only read off a template strand of DNA. So the template strand is the 3' to 5' strand. Just like DNA polymerase, RNA polymerase reads the strand in the 3' to 5' direction. When it does that, it's going to add nucleotides on, and each nucleotide that is added in the 5' to 3' direction. So these two are anti-parallel to each other. So my new piece of RNA that I'm making is 5' to 3'. All right, and then the last step is called termination. So that means ending. So when an RNA polymerase reaches a specific sequence, it's actually going to stop. It's going to terminate, and it's going to cut it off, and then the segment is free to go. So I really liked this picture, although there's a lot of wording on it, but it shows that there was a promoter region right here with a TATA box, and that's where my RNA polymerase joined, so that was initiation. My template strand of DNA is my 3' to 5'. My new nucleotides, so we get RNA nucleotides from the food that we eat. When we are eating things that were once alive, they have DNA and RNA in them. So the RNA breaks into all little nucleotides, A's, C's, U's, and G's, And then those are used to assemble this new RNA transcript. So here's elongation happening, and it's making a 5' to 3' strand of RNA. And then eventually, this is showing elongation, but eventually it's going to reach a sequence here that's going to be this termination sequence. So when it hits this spot, it's going to let go, and this orange RNA transcript is going to be free in the nucleus. All right, the next thing that happens, first of all, we do not make just one kind of RNA. We actually make 3 different types. The first one is our messenger RNA. mRNA stands for messenger. That is going to be modified. It's going to leave the nucleus. It's going to go to the ribosome, and it codes for a specific amino acid sequence. So to build a protein, you need amino acids in a specific order. The messenger RNA is what really decides that order, and it's all based on the DNA. All right, the next one is called transfer RNA, tRNA. Transfer RNA folds into this cloverleaf shape. It leaves the nucleus, it picks up the amino acids, and then it joins with the mRNA at the ribosome to actually build our protein. The last RNA is called rRNA. R stands for ribosomal. So ribosomal RNA, it combines with proteins and it forms this globular mass called a subunit. There's a large subunit and a small subunit, and those will come together to form a ribosome in the cytoplasm. All right, then the last thing we need to talk about is our RNA that is messenger RNA actually gets modified before it leaves the nucleus. There's 3 steps to the modification of RNA. The first is you got to protect it. So it's about to leave, and remember we talked about the cytoplasm being this liquid that has enzymes in it, and it could potentially be dangerous, and it could break apart my mRNA. So it's sort of like putting little anglets on a shoelace. It protects the 2 ends. So the one end, which is the 5' end, that's going to get a methylated cap. A guanine triphosphate is added to that end, and it methylates it. It protects it from things destroying it. The next is the 3' end. That gets what's called a poly-A tail. So poly means many, A is adenine. So you add many adenines to the end of the 3' segment, and that protects it. The last step is called splicing. So there are actually sequences in our DNA that do not code for anything. They certainly make us unique, and you can tell different people's DNA by their different introns. But those non-coding sequences have to be cut out because they don't count. Think of introns as being in the way, and think of exons as the ones that need to be expressed. So we need the exons to come together, and we need the introns out. So that's done by small nuclear ribonucleic proteins, SNRPs. These are called SNRPs. And SNRPs together make up a spliceosome. So I really liked this picture. So, except for it does have a mistake. This should, if this is my DNA, this should be 3' and this should be 5'. All right, so here's my original DNA with my promoter, and it's made into this messenger RNA, and this messenger RNA is going to be 5' to 3'. Here's my cap, here's my tail, and you'll see that there are green exons. Those are what I want. I want to express my exons. My introns are in the way, so a SNRP would cut these out. It actually makes this, it's called a lariat. It's like a little loop, and it cuts them out. And when they're cut out, this is spliced together. So when it's spliced together, I now have my cap, I have my tail, and I have my exons. So this is ready. Here's a picture showing those lariats. So here's a SNRP. Here's a molecule that's going to join. It actually cuts this and bends it. So what it does is it bends it over, makes a loop, cuts it off, and then attaches it to the other sequence. And so this is the part that's going to be degraded. This was the introns that you don't need, and now the exons get spliced together. All right, so I want to make sure that you go back over this, look it up in your textbook. There are great pictures, there's great information, and then go find some Google images and test yourself. See if you can explain this to somebody else. Could you draw it out and understand what happens as mRNA is made and modified?